Good afternoon, everyone. So my name is Sharon Pepinella, and I am one of the managers of the Barcode Long Island program. And on behalf of the entire Barcode Long Island team, I would like to extend a warm welcome to our mentors, esteemed guests, and of course, our talented young researchers as we gather here for our 9th annual Barcode Long Island Student Symposium. Today, we celebrate the power of knowledge, the beauty of scientific discovery, and the remarkable achievements of these young minds who have delved into biodiversity research through the process of DNA barcoding. This symposium serves as a platform to showcase their innovative projects, their unwavering commitment to scientific excellence, and the relentless pursuit of understanding the world around us. The posters developed by our students, including those that will be presented today, as well as posters from previous years, are available to view on our DNABarcoding101.org website. Here, you can also find information about our different DNA barcode programs and other available resources. So as many of you know, DNA barcoding has roots at Cold Spring Harbor Laboratory, where in 2003, the first barcode meeting was held at Cold Spring Harbor Laboratory's Banbury Center. Attending scientists were wondering if it were possible to use a certain region of DNA to identify an organism in the same way in which a supermarket scanner uses a UPC code to identify a product. It turns out that not only is it possible, but it works pretty well. Shortly thereafter, the DNA Learning Center began developing tools for student-driven DNA barcoding research. In fact, next year will mark the 10-year anniversary of our Barcode Long Island program. DNA barcoding has been used as a transformative tool in the field of biology for the past 20 years. Through DNA barcode analysis, we can better understand and restore ecosystems, monitor the loss of biodiversity, slow the spread of invasive organisms, survey vectors for disease, track the movement of species, monitor the effects of climate change, combat poaching, and more. Additionally, DNA barcoding is being used to provide governments around the world with information to create better policies for environmental preservation, to guide health regulations, and to protect consumer interests. With us today are the students and mentors representing the 61 DNA barcoding and metabarcoding projects conducted by these talented students over the past year. We are thrilled to celebrate the efforts of these participants as their discoveries go beyond the classroom, contributing to our collective knowledge and pushing the boundaries of scientific exploration. Students, your dedication and perseverance make you truly great scientists, and you should be extremely proud of your accomplishments. And of course, we would like to extend our gratitude to the mentors, families, and supporters who have guided and inspired these young researchers. Your expertise and unwavering belief in their abilities have played an instrumental role in their journey. Your commitment to fostering a love for science among the next generation is truly commendable. I'd like to say a big thank you to the Barcode Long Island team, especially Dr. Cristina Fernandez-Marco, the co-program manager of Barcode Long Island, as well as the entire DNALC staff for all of their hard work in the continued development and successful execution of our DNA barcoding programs. Thank you to all of our collaborators who've continued to support our program in numerous ways. And finally, a big thank you to our original funders who allowed us to develop this sustainable research program. It is now my privilege to introduce to you our keynote speaker, Dr. James Lendemer, Associate Curator at the New York Botanical Garden and Adjunct Assistant Professor at the City University of New York Graduate Center. Dr. Lendemer received his bachelor's degree in biology from the University of Pennsylvania and his Ph.D. in biology from the CUNY Graduate Center. His work with natural history collections began in high school, and his appreciation of history and collections is what led him to build a career out of collection-based biodiversity science. Dr. Lendemer has studied lichens for over 20 years and received multiple awards for his biodiversity-focused research that weaves together data collected over centuries with extensive new work from the field and lab. His most recent research efforts are increasingly in the context of responses to change, such as through human disturbance and alteration of the environment, or long-term geological processes. We are excited to have him with us here today. Please welcome Dr. Lendemer.
Like it. Can people hear me? We're all good? All right, great. Just see if we can get this going. All right. Lights are down. The deluge continues. Great. So let's get started. So I want everyone to imagine an alternate world, a world where everything, trees and rocks, bricks, seashells, even the soul is completely cloaked in an intricate fabric that's living and made up of these beautiful colors and infinitely complex patterns. So across environments, forests and cities, natural and built, this fabric is macroscopic. It's readily visible, but it's almost impossible for anyone to really truly see and to appreciate fully. There's grays and blues and yellows and oranges, reds, greens, and browns. It's a fabric that's made up of fungi forming communities with each other and with a diverse array of partners that have contrasting, complementary, and at sometimes antagonistic purposes. Collectives that are so strong that they influence the environment and so important that they influence everything around them and interact with everything around them. Birds and moths, seedlings, and human cultures. Everything has integrated them into aspects of their own experience and their struggles to advance and to survive. Communities that are so time-tested and resilient that they've evolved over millions of years to live in the most extreme environments on Earth, adapting to ever-changing climates. But at the same time, so fragile that they can't be easily moved. They're easily damaged and completely unravel when they're confronted with rapid changes or with pollution. A fabric that humans perceive as static and unchanging, but it's really abuzz with this hum of birth and growth and death and competition to survive and collaboration to colonize, spewing swarms of spores and other particles into the air that we breathe. We're all breathing them right now. These remarkable fungi are awash in contradictions. They're strong, but they're weak. They're competitive, but they're collaborative. They're internally frenetic and dynamic, but they're also superficially fixed and permanent. They're seen, and at the same time, they're unseen. That's lichens. So thanks again, everybody, for coming today. And I'm James Lendemer. I'm the lichenologist at the New York Botanical Garden, and I wanted to tell you a little bit about scratching the surface of some of the dimensions of lichen diversity. So before we get started talking about lichens and fungi, I wanted to step back a little bit and think more about the history of study of these organisms, because it really gives you a little bit of a perspective on why they have ended up being so sort of understudied and neglected, and why they require so many methods now, such as DNA barcoding. So who here has heard of the word cryptogam? Raise your hand. Almost no one. That's every audience that I speak to, believe it or not. Well, what the word cryptogam means is it means hidden sex or hidden gamete or hidden marriage. And this word is used to collectively refer to all of the botanical things, plants, fungi, and animals that used to be classified with plants and fungi, even though we know they are not those now, but all these things that reproduce with spores rather than seeds. And the reason that I say this, and I even introduce this word besides giving you something new and fun to say to your friends, is that this really explains that these things are unrelated organisms that were grouped together hundreds of years ago by the people who first started studying them, because they couldn't actually understand them. They couldn't actually understand how they reproduce. They couldn't see it, they couldn't understand it, and so they just grouped all these things together. And so what we have are things like ferns and mosses and liverworts and lichens. Those are all cryptogams. They all reproduce with spores. They were all classified historically as plants. And now we know that they're not necessarily all related to each other today. So that's cryptogams. Take that with you into the world. But maybe a little bit more important than that, fungi are cryptogams. Now there's millions and millions of species of fungi on Earth. They're one of the most diverse life forms out there. They take on every form, every color, every size and shape imaginable. To me, fungi are absolutely amazing. But what's really, really remarkable about them is that they span all the possibilities of the spectrum of life, right? Some of them are pathogenic, they kill things. Some of them are beneficial. Some of them are commensalistic, some of them are symbionts. They do all sorts of different things and they're ubiquitous in our environment. They're also taking on all of these colors, all of these shapes, all of these roles, but yet somehow entirely made up of these basic fundamental units called hyphae. Fungal hyphae make up every bit of the fungus that you see, and all that they are little strings that are woven together in different ways. To me, that's amazing. If there's nothing else you take away from today, it should be that fungi are absolutely amazing because they're made up of such simple things that have such a huge impact on the world and are so diverse, visually and biologically, evolutionarily. Fungi are incredibly important, and they are incredibly important because of the relationships that they form and the relationships that they're involved in. And if there's one group of fungi that I suspect many people here might have heard of, it's mycorrhizae. Raise your hand if you've heard of mycorrhizae. They're more than cryptogams, see? Mycorrhizae are these fungi that form this imperceptible network among living trees and shrubs and other plants. 80 to 90% of the plants on Earth form these associations with these fungi, and we would not have the bulk of the vegetation around us if it were not for these things. Many of our most important agricultural crops also associate with mycorrhizal fungi. So these fungi enter into mutualistic symbioses with plants. That means that both of the partners in this situation, the fungus and the plant, get something beneficial out of the deal. So they're fungal mutualists, they're mutualistic symbionts. And these associations are incredibly important. Another kind of mutualistic symbiosis is lichens. And lichens are, if mycorrhizae are sort of like the thing of the moment, lichens are going to be the mycorrhizae in 5 years. I promise you that. So whereas mycorrhizae form these like networks in the soil linking everything together, lichens are these completely self-contained units of all sorts of living things. And the really remarkable thing about them, well, there's a million remarkable things, which I will touch on today, but one of the most amazing to me is that every single time you see a lichen species, which is a species of fungus, it looks the same. And they form these very complex structures. Maybe they don't look particularly complex to us, but I think it's pretty amazing that this thing grows like this in nature when you consider that it's actually formed by multiple constituent parts. If they were to grow on their own, the fungus would look like shower mold or mycelium, bread mold, something like that. Doesn't look like much. Don't tell any mycologists I said that. If their photosynthesizing partners were to grow on their own, they would also not look like very much. Don't tell anyone that studies cyanobacteria or algae that I said that. But yet somehow, these two things that look like basically smudges or little, patches of felt or something on rotting wood or on trees or rocks, somehow when they get together, which they have to do for the lichens, for the species that form lichens, they form these really complex structures that are unlike anything else in nature. And to me, that's completely amazing. And not only do they do that, they don't just do it randomly, they do it all the time. Every time the fungus grows with its partner, it looks the same. And they have to do this to survive. And so what you end up with are these complex structures where you basically have layers of purely fungal tissue that are enclosing a layer that has the fungus plus its photosynthesizing partner, which is providing it with nutrition, with the food. It has an internal food source. In most cases, it's a green alga. Sometimes it's not. Sometimes it's a cyanobacterium. But regardless, it's a photosynthesizing microbe. So lichens are these symbioses formed between fungi and algae. And when we talk about lichens, we're talking about these species of fungi that are entering into these interactions. Every time you see a lichen that looks maybe like this, let's just say, this lichen is called Anaptychia palmulata. The reason it's called this is because every time you see this lichen and it looks this way, it's this species of fungus forming this lichen. Now, historically, the way we thought about symbioses in nature was that they were formed by consistent, typically consistent, one-to-one relationships between species. And so when I learned lichens 20 some years ago, my understanding was that every time you see this thing, it's this fungus, Anaptychia, growing with this alga, Trabuxia glabra. I actually made that up, but let's just pretend that's the actual name of it. So that's how we conceptualize them. But then, and you have to understand these algae, if anyone here has looked at them under a microscope, they look like tiny green balls. They do not look like very much. And it's very difficult to tell the difference between different species and especially between different closely related species. So with the advent of DNA sequencing, with regular Sanger sequencing, people started to sequence the DNA of the partners of these lichens. And they found out, oh, wow, it's actually more complex than we thought. It's not every time this species of fungus grows and makes this lichen, it grows with this other thing. But instead, maybe every time it grows in New York, where it's like a pretty temperate place, we've got cold winters, we've got hot summers, maybe every time it's in New York, it's growing with Anaptychia with this Trabuxia glabra. But then maybe, I don't know, in Florida, let's just say, where it's a lot less temperate and it's more subtropical, where you have warmer summers and not as cold winters, maybe this fungus associates with this other partner that's better adapted to the climate of Florida. Maybe we couldn't tell that before because the two species look really similar. But thanks to DNA, we could figure that out. Then let's just say maybe you see the same thing growing in, I don't know, Kentucky, and it associates with a different partner that's better adapted to the climate there. Okay, so the situation is more complex than just a one-to-one thing. It's like actually this fungus can associate with different partners, and it's different partners that are better adapted to different locations, different climates. That makes a lot of sense, right? That totally turns the head on how I learned this, incidentally. The first thing somebody asked, when I first started studying these, it was like, what about the algae? Nobody looks at those, we don't care. And it turns out that they're actually what's driving everything in the lichens, because they're what determines whether it's an environmentally suitable location and the lichen can grow there. But wait, there's more. So with the advent of DNA metabarcoding, what we found is that all those different things can actually be in one individual. And so rather than a one-to-one relationship, and it's just sort of different everywhere it's growing, actually when you see a single lichen, it could have this whole pool of partners, and those different partners have different suitabilities for different environmental conditions. And that's, we think, how these things change over time and adapt to changing climates, changing conditions. It's a very slow process, but we think that that's how that happens. So then what is a lichen? If you look at one individual lichen, this is what I think most of us would look on if you looked at a tree or you looked at a rock and you saw this one little splotch on there, you'd all probably say that's one lichen, right? It's an individual, just like one cat or one sunflower plant. Can I get some nods? Yes, right? We'd all agree it's one individual, it's one thing, right? Okay, well, we know that actually this thing would have multiple different partners that are photosynthesizing in it, so different algae, and there'd probably be some different genotypes of those algae, so different individuals of the same species of the algae in there. We also know it's got a whole host of microbes, a microbiome, just like we and everything else on this planet does. Some of those things are good, some of them are bad, some of them are kind of like, meh, they're there. But then also the fungus that makes up that dominating thing in the lichen can be present as more than one individual as well. So what we perceive as a single individual is really a community made up of multiple individuals of all of the different partners and of just other stuff as well, growing together to form what we perceive as this one static, unchanging thing. Minds are blown. So then what is a lichen? A lichen is actually a community. So you could take this lichen, this is candle flame lichen, Candelaria concolor. It's really common around here and in New York City. And you would have your dominant fungus that makes up the lichen. And then you have its photosynthesizing partners that are providing it with its nutrition. And then you've got all of these microbes inside of it, different bacteria, viruses, things like that. Some are good, some are bad, some are just kind of hanging out there. And then you have all sorts of other fungi that are not the lichen growing in there as well. You have little invertebrates growing inside and around the lichen. And then you have a whole world of chemicals that are involved in signaling between all of these different things, and also trying to keep some of them at bay, help some of them out. They have all sorts of different functions. But it's amazing to me that you can imagine all of this is happening all within one little splotch on a rock or a tree while you're looking at it, and it looks like nothing is changing. But then imagine what's happening at the scale of when you have different individuals, what you perceive as individuals, growing together next to each other in a community, right? And then also when you have different species of lichens growing together in a community. You have all of these different communities of different species of fungi, different photosynthesizing partners, different microbes that are all adapted to the hyper-local places that they're living. And all of those partners are potentially being shared amongst different individuals of the same species and amongst different species in the community. And so many of the members of the community are actually dependent upon each other for their collective survival because they're sharing all of these partners, and the partners are locally adapted to where they live. If you look at the same lichen species growing in two different places, a mile apart or 2 miles apart, they could have very different internal communities, but those communities would be shared by the other lichens that are growing nearby. We had no idea that this was the case 20 years ago. In fact, we really didn't understand that this was the case until relatively recently. It's really next generation sequencing that's allowed this to sort of like become a clear thing. And so lichen communities are really these amazing dynamic things, and they're these amazing dynamic complex sort of associations of interactions between lots and lots and lots of different things. Like, it's incredibly meta. And so the way we think about life traditionally has been to think about individual species or individuals of species in isolation. So there's millions of species on Earth, right? There's all sorts of beautiful things out there, moths and fungi and birds and newts. But we've traditionally thought about them in isolation, in sort of isolation from each other. But what's become increasingly clear, relatively recently in science is that it's not just, you can't just study something on its own in isolation, but rather that the key to understanding life on Earth in all its diversity and all of its complexity really relies on understanding the relationships between different species, and different individuals. It's all about interactions. And those interactions can be biotic, so they can be among living things, and they can be abiotic. They could be between living things and the environment. But the reality is that so much of science now has flipped on its head and focused on interactions. You can even see it in nature documentaries. If you look at, if you watch a David Attenborough documentary now and you compare it to one that came out 10 years ago, everything now is about the interactions between organisms. And like, the stories are amazing. I mean, it's the kind of thing that you would not believe that it exists, but then it's like, oh, actually it does. And the interactions are what's so key to the success and the survival of so many species. And it explains so much of what's going on, the patterns and processes that are underpinning everything that we see in the world around us. So if interactions are important, what do you think is so hard about studying interactions? Someone? Imagine. Yeah, go for it. Like, what do you mean? And it would be hard to observe, right? So imagine, you're right. So you can't be sure, how are you sure that you're always going to see this thing that's going to happen versus something that's very aberrant? And then also imagine that, it's easy to study maybe two interactions between two different things, but then what about actually all the things that are growing around them interacting with each other and themselves? It's like basically impossible, right? Imagine like all the birds in a forest, and then you're trying to study how they're all interacting with the plants and everything that they're feeding on and all the insects, it quickly becomes essentially impossible. Well, that's why lichens are amazing. Because lichens are these self-contained systems of interactions, and the interactions have to take place to get the lichens. So if we really want to understand how it is that these interactions underpin so much on Earth, the lichens are the ideal way to do that. And so we've started to do this. And so what I want to start on talking about today was, rather than begin in cities, I wanted to take you on a story of the wilderness to kind of get a sense of exactly how it is that these interactions are so important to the lichens, but then also what that potentially means more broadly for the environment. So I work a lot all over the Eastern United States. And for the last more or less decade now, I guess, I've worked especially in the Appalachian Mountains. So the Appalachian Mountains are the main mountain chain here in Eastern North America. And if you want to study biotic interactions and how they or interactions in general, and how they influence patterns and processes in nature, well, lichens are a really good way to do that. If there's a place to do it, it's the Appalachian Mountains. And the reason for that is that, besides that they're very scenically beautiful, it's that they have these amazing stark gradients of like extreme elevational changes. You can go from like under 1,000 feet to over 6,000 feet in 5 or 6 miles. You have extreme gradients of precipitation. So some places are super wet, they're rainforests. Some places are much, much drier. And those places can be right next to each other on a mountain, or on different faces of a mountain. So you have these really extreme environments, and you also happen to have these really, really, really diverse ecosystems. There's more species of trees in one national park in the Appalachians than in all of Europe. You know, there's so many plants, there's so many animals. It's the salamander diversity capital of the world. The Appalachians are amazing because they have all of these different things going for them as the ideal place to study this. And so what we did is we focused on the Southern Appalachians, which are a region from Virginia to Alabama. It's the highest amount of diversity in the Appalachian Mountains and one of the most biodiverse places on Earth. And so we went through this region and we cut it up into bands that were 100 meters in elevation. And then we also looked at every aspect of the mountains. And we established a series of 1 hectare plots all across this area. And then we put these plots within that framework. So every 100 meters on every face of the mountain. And then we coupled them for places that had been disturbed by humans and not disturbed by humans, or actually disturbed by various forces, disturbed and undisturbed. And then within each one of those plots, we did a lot of different things. You can just see there were 208 of them, incidentally, in the end, spanning from basically Southern Virginia through Alabama. Within each one of those plots, the plot was unique. The plot was standardized in that it was one vegetation type. So we weren't crossing lots of different things, right? We were staying in an oak forest, we were staying in a heath ball, we were staying on a granite dome, something like that. And within each one of those 1 hectare plots, we spent a lot of time identifying and collecting all of the lichens that occur there. And we had to collect them and bring them back to the lab and identify them, because you can't be absolutely certain, as I'm sure some of you found, of what they are in the field. And maybe it's not even so certain when you bring them back to the lab. And then we also assessed the habitat quality of all of these plots using a standard set of metrics that we developed out of a set of forestry habitat quality metrics from the Pacific Northwest. We also measured the DBH, the diameter breast height of all of the different trees in a smaller area of the plot. And what that does is it gives you a sense of how big all of the trees are, and then also how differently sized they all are. That tells you a lot about the age and the history of the forest. We also quantified how loggy it was and how rocky it was. Those are really important factors, we thought, potentially for lichens. We also we quantified all of the species diversity for woody plants, so all the trees and all of the shrubs in all of those plots. You can imagine this took a lot of effort. It was really fun. And then we also went and we collected environmental DNA, eDNA samples from trees and rocks all over in these plots to try and get a sense of the spore bank. So we did a lot of DNA metabarcoding here. And I won't be talking about that in specifics today, but I will tell you that the DNA, the spore bank is dominated by propagules of lichens from the local area rather than things from far away. Something that we actually did not really know before. So this whole area, we surveyed all these plots, and what we found is that there's about 1,200 species of lichens in this area. That's a pretty good number. And in our plots, so that's sort of overall plus literature and all this other stuff. But then we found out that we captured about 75% of them in the plots that we studied. That's actually pretty darn good. We did good. A lot of those other ones that we missed are things that were hyper-localized or had been extirpated because of air pollution. It's one of the most, it has had some of the heaviest air pollution in the United States in the past, or because of past land use, past deforestation. So, you know, in the end, not all of 1,200 things that could be there were there still. And in the end, we found 872 of them, which is actually pretty good. Here's a map where they were of these 208 plots. And here is the number of lichen species that we found in each of them. Now, this alone is actually a really cool thing to see because you can, if you just look at the colors, you can see that the high and low diversity places are not all concentrated in one place. There's really high and really low diversity places right next to each other, which is like, oh, wow, well, if we want to understand what is underlying these patterns of diversity, well, there's a good way to, you know, this is helpful, right? This is actually also a very different picture than what we have here in the mid-Atlantic region and in the Northeast in general. All of our highest diversity areas are all concentrated in a few small patches of forest. So this is a very different situation there. And in the end, we focused on just what was in the main part of the mountains because all of those other plots in Alabama, further south, are very different. They have very different communities of lichens, and they're different environmentally. So in the end, we focused on just this smaller subset of 161 plots. And we had a lot of data. All these variables, you can only imagine the spreadsheets. We have all of these variables on environment. We have all these variables about the rock and the habitat quality, the tree diversity, all of these different things, the partners of the lichens. And in the end, so my colleague, Christine McCain, was the person who led this part of the project. In the end, when you analyze all of that, what's the single biggest driver of lichen diversity in this region? Lichen species richness. It's habitat quality. That's not a huge surprise because lichens are bioindicators, and anyone that's been to a city will tell you that there are fewer lichens there than outside of the city. But this has actually never been shown before, and certainly not to this degree and in this way. But then we were curious, okay, so great, big whoop. Habitat quality drives diversity of species of lichens. That's not a big, amazing new discovery. It is important, but it's not that new. But then what happens when you take habitat quality out of the equation and you only look at the sites that actually have a higher habitat quality? So like 60 to 100 means that it's like a decent forest. Like you'd walk in there and you'd be like, oh yeah, this is a nice place to hike. Well, when you do that, you take habitat quality out of the equation, it's no longer significant when you look at them over that level of 60 to 100. Again, it's just a score of 0 to 100 is habitat quality. So you take your 161 sites, we reduced it down to 132. Doesn't actually look like that big of a difference, right? Barely noticeable. So much work away. When you reanalyze everything and look at all these variables, what we see is that the single biggest, the only significant things that come out of this all have to do with living things. So species diversity of lichens is highly correlated with the number of tree species, native tree species that there are in a stand of forest, and also with the canopy cover of native tree species in the forest, which has to do with sort of age and history of the stand. And so this is actually really important because it means that it's these external biotic factors that are driving the richness of these species. So it's living things that have to do with this, right? We didn't know that before, and we certainly did not know that before at this scale. So this tells you that if you care about lichen diversity, having a good diversity of living woody substrates that are native means a lot to that. And so it's these external biotic interactions with living things that are so important for the lichens. And that complements something that we already knew from what I told you earlier, which is that the internal relationships with these photosynthesizing partners are also so important, right? It's the partners that are driving where the species are able, or where the lichens are able to grow, not the fungi. The fungi can grow all over. It's the partners that can't. So there's three main different kinds of partners that lichens associate with. You don't have to pay attention to what they are. They have these names. They look a little bit different. The lichens they make look a little bit different. But you can even see, just looking at the pattern of their species richness with elevation, that they're completely different for the different groups. So it's interactions with living things from the outside that drive patterns of lichen species richness or lichen diversity. And it's also patterns, it's also relationships or interactions with things inside that are doing that. It's all about living things. It has very little to do with the environment, directly at least. And that's really important because so much of our understanding of biodiversity patterns on Earth pertains to plants and large animals or vertebrates. And much of what drives their patterns of species richness has been thought to correlate with climate and with the environment. But it looks like maybe, it's actually interactions with living things that are potentially having a bigger role. And at least when you're able to study them in this really constrained way, you can see that. One of the cool things about a project like this, by the way, and this goes anywhere, is that when you get to know a region in depth, when you get to study something kind of really in depth, you find things that you would never have normally, you might have overlooked before. And you can find things that are actually really important that you overlooked before. And an example of this is this species, Shadonia sols kellii. You can excuse the name. It's named after a pioneering environmentalist in North Carolina who stopped clear cutting in the Southern Appalachians. This species was previously unknown to science. And it turns out that it's actually the dominant lichen that grows in spruce fir forests, which occur at the tops of all the mountains. And those are all critically endangered due to climate change. They were reduced to 99% in extent by logging historically, and now they are on their way out thanks to climate change. Well, these dank forests that are just laden with mosses and lichens, the dominant lichen was something that was not known to science until this year, or not described to science until this year, which is crazy to me. So this kind of thing is needed everywhere. It's needed here on Long Island, it's needed here in New York City, it's needed everywhere. And the reason for that is that we don't have a lot of great modern data, something that I suspect some of you also have learned about during your projects. So this is Trenton, New Jersey. And these yellow dots are everywhere where there were historical collections of lichens from Trenton and the surrounding area. I forget what county that is. There were only 55 collections of lichens made here in all of time prior to like 2020, 2018. And most of them were misidentified, long story. But the point is that then my colleague Dennis Waters went out and he conducted a really detailed inventory of all the lichens in and around Trenton. And he found almost 200 species, and all of them, this is like a completely different picture, right? So not only can we try and understand how things have changed, like are those 55 things or whatever were there before still there, but now we actually have this better picture of what's there today, and we can try and understand how it relates to humans, human change to the environment and the environment in general, as well as to all the other lichen species there. And so what I'd like to move on to is New York, to our local area. And this certainly includes Long Island. So New York City and all of Long Island once had these really thriving lichen communities of really, really diverse and beautiful things. These species all occurred here, and we know that. How do we know that? We know that because historically they were documented by naturalists. So we know, for instance, that the lungwort, which is a big lichen that grows about this big on trees in old growth forests nowadays, it's been lost from most of its range in the eastern United States. We know that this species used to grow in the Hamptons in the 1890s. Keep in mind, this is a lichen that's this big. Any of you that did projects on lichens, you can appreciate the size of this thing, right? Like you probably didn't see anything quite that size. Looks like a lettuce. This one over here, this is from the Palisades of New Jersey across the river from Manhattan from the 1860s. No such thing grows in the Palisades of New Jersey now. So we know that the lichen communities in this region were once really, really diverse, just like in the Southern Appalachians. And we know that because of these collections. And we know that the primary reason why they're not diverse anymore is because of disturbance and human change and habitat quality change. And we know that because we've done this work elsewhere. And it's certainly the same situation here. But the story of New York City is really interesting. And the New York region in general is really interesting because we have one of the best documented lichen baselines of anywhere in the world, certainly anywhere outside of Europe. And it's because by pure chance, there just happens to have been someone that was crazy enough to study lichens in this region about every, like 50 years or so. And so we know that in the 1820s, within 10 miles of Lower Manhattan, of the City Hall in Lower Manhattan, there were over 190 species. And then we know that by 1914, there were far fewer. By 1968, a graduate student from the New York Botanical Garden extended that radius out by 50 miles from downtown Manhattan because he couldn't even from City Hall, because he couldn't even find any lichens in that area. So he only found 6, and that was within 50 miles. So a much bigger area. And then more recently, what we found is that there's over 100 now. And so you see this pattern of sort of decline and then improvement. What happened? What happened in our region is what happened everywhere else. New York City and our metro region built out, the grid was introduced, and large areas of what were natural habitat and agriculture were converted to urban area and suburban area. Well, suburban, but urban. But during that process, there were still, fence rows of trees. People still had gardens. There was still parkland and things like that that had some remnant of what was there before. And so that's why some lichens were able to survive that, probably a few more than the 51, that were there in 1914. But then air pollution came into the mix with industrialization, and there was some of the worst air pollution was in New York City. And that is what got rid of almost everything else. We know that by the 1960s and 70s, there was not a lichen in New York City at all. And there were very few going out on Long Island. The further you went out towards Montauk, the more species you picked up. And we know that because somebody actually studied that in the 1960s. And there's an amazing foundational work on the lichens of Long Island that could be repeated now and needs to be repeated now. And then now we've had the Clean Air Act improved air quality. Sulfur dioxide is less in the air now. Sulfur dioxide kills the partners that the lichens feed on. So their partners internally that provide the food, they die from sulfur dioxide exposure. So then they end up the fungi starve to death. It's kind of like coral bleaching. We have cleaner air, so that's good. We also stopped building giant cities. And so the environment is more stable. So we have a cleaner environment and it's more stable. And what that has meant is that lichens have been able to recolonize, or at least some of them have. And you can see that everywhere you look. So this is just a comparison of a couple places that were inventoried in the past. The High Line in Manhattan, Alley Pond Park in Queens, the Brooklyn Botanic Garden in Brooklyn. And you can see that everywhere that's been looked at more recently than in the past, more species have turned up. And that's just a really coarse way of saying there's more stuff here now. And we know that it's because of improvements in air quality and having a more stable environment. And so if you look at the lichens that are around us here now, they fall into 3 main categories. Or sorry, if we look at the lichens that have been here at all, they fall into 3 main categories. A lot of them have been completely lost because of that changes in habitat quality, air pollution, things like that. A very, very tiny number of them persisted. Further out from the city, way out on Long Island, more of them persisted. But some persisted, but it was a relatively small number compared to what was here originally. And then by far, the majority of them have come back. But how have they come back? They've come back by adapting to the changed environment. That means they've had to find new partners that are suitable to these environments. So most of them have actually returned. And what's really interesting about that is that they're returning, right? But they're returning in kind of this really thoughtful, thoughtful way. And so an example of that is Usnea mutabilis. This is an old man's beard lichen in all its glory in Maryland. This is what it looks like when you find it in New York City. So it's been gone for well over a century. And now we've found two individuals, one in Woodlawn Cemetery in the Bronx, one in Greenwood Cemetery in Brooklyn. Those two individuals were so small that they could only be identified with certainty with DNA barcoding. And that's going to certainly be the hope for many of these lichens that are returning to areas where they weren't before, because they're going to be small and they're going to be hard to identify because they're growing in harsher conditions than they're used to. And so, you know, this is an example of a species that's returned, and it's returning to these really stable environments. Cemeteries are places that people don't really build lots of new things in and generally don't disturb very much. And they tend to just kind of have a lot of like nice greenery and stable environments in general. But what's really interesting is, you know, we transplanted the same species to the New York Botanical Garden in 2015, and you can see what it looked like by 2017. So it turns out that you can't move them. And by far, most of the lichen transplants that have been done have not succeeded all that well, certainly not in our area. And that's partially because when we move them from a place that they're suited to a place that they're not. And that's because of the partners, those internal associations, interactions with living things. And so that's why they have to come back on their own. And so you can study this, anyone can study this. There's a whole bunch of folks have gotten really into this in New York City. You can look at trees that are moved in, right, planted anywhere, especially on campuses of schools. This is a really big street trees and campuses of schools. Here's a tree that was planted, an ornamental maple that was planted in front of an apartment on 22nd Street in Manhattan in 2015. You can see it's covered in lichens. The tree probably came from Long Island based on the lichens that are there. You can see what the lichens looked like in December of 2015. They were kind of like not so great. They're a little sad, womp womp. But then by 2020, they're completely gone. But that's not always the case. So sometimes you move lichens to a place that's actually suitable for them. So for instance, here's some trees that were planted in Rockaway, Queens in 2022, and the lichens are doing perfectly fine. Several of these are species that do not grow within hundreds of miles of New York City naturally. But we're now finding them on planted street trees all over because air quality has improved enough that they're able to survive now when trees are planted. And a really good way to think about this is the High Line in Manhattan. So the High Line was inventoried many years ago before it was converted into the park that it is today. Like hundreds of thousands of people probably visit it every day. I don't know the exact number, but it's a lot of people. So most of it used to look like this abandoned train line here, right? With gravel and stuff on the bottom. And there was one lichen growing there, I think in the 90s, or maybe it was the early 2000s. And so I was asked to come and inventory the lichens there. And what I found was that the only place there were lichens on the High Line was in the one small area where the old railroad bed remained. The entire rest of the converted park, which is planted with these beautiful plantings and trees and all sorts of things, has no lichens in it. But yet, you know, above everyone's heads in Midtown Manhattan, there are over a dozen species of lichens that have managed to return naturally. So they've managed to adapt to the environment, and they've managed to establish and live and thrive in this one little patch of railroad that remains. And so that's actually really important, because that means that these are the ones that are likely to survive to the future, you know, in the future conditions of this planet, right? A planet that's more urban, a planet that's more sort of stressful for these things. These have found partners, they've managed to adapt, and they've managed to survive. But if we convert this last little piece of the High Line to the rest of the park, we'll lose these species that have managed to adapt to the conditions of Midtown, which are extremely stressful for anyone, I think. And so, you know, what I would just sort of like to end with is the fact that understanding the relationships between all the things inside the lichens, but also just of all the living things around us in the communities, natural and not, is incredibly important. And it's important to really focus on the relationships between these different things, because they have really important implications for ecosystems. And understanding those relationships, what's there and how they interact, is really important, not just for like the ecosystems themselves, but because these places have already changed dramatically from the way they were historically, but also now they're rapidly changing as well. So it's like change on top of change. And so if we're really going to try and conserve biodiversity and, continue to have nature around us in some form in the future, so much of it really relies on understanding the biodiversity around us. And that's what y'all are doing today. Thanks.
Okay, so we have about 5 minutes for some questions. I have a microphone that I can pass around, plus there are some microphones on stands along the center wall. So if you have a question, you're welcome to go down and ask into that. Does anybody have any questions for Dr. Lendemer?
There should be so many questions. I have lots of questions for me.
It's not really a question, but more of a comment. It's like you've kind of given me a newfound appreciation for these little colonies, because originally I have just thought of them as these strange little patches on trees, and I kind of thought them as of just a normal occurrence. So learning that in like places like New York City, they're so scarce, and yet they've somehow managed to return despite the harsh conditions, I've definitely gained like a new appreciation for these extremely diverse colonies.
Yeah. That's great. And I hope everyone else feels the exact same way. Because, again, it's not just New York City. What's so important is to remember that this is everywhere. The lichens are a very good way to understand how the world around us has been impacted by our past activities and how it's likely to look in the future. The lichens just happen to be this really convenient lens to view that through, because they're so susceptible to change that like you can readily see it. When they are disturbed or impacted, they disappear. They're not replaced by something else. They're just gone. And so the return is a really important thing, but it's also important to remember that as these things return, what's coming back is a small, tiny subset of what was originally there. And it's also, the function of these things in the environment is so incredibly important. And the question is, how has our environment, how have our ecosystems changed from a functional standpoint by the loss of all of these things? Everything should be covered in a coat of lichens and mosses, and it is not anymore. And what does that mean for seedlings? What does that mean for invertebrates, let alone and trees and animals, let alone the lichens and the mosses themselves? Which I think we should care about maybe more than all those other things, but that's just me. So thanks. Yay.
If they're gone for so many years, where do they return from?
They very, very slowly return from the nearest possible location. So like what we learned from our eDNA barcoding work, which was led by Nolan Kane's lab at University of Colorado and former graduate student Kyle Keepers, what we found is really that the local environment is dominated by spores and stuff of the lichens that are right there. And other studies have like captured spores and stuff in the air and determined that basically all of the spores and the propagules that would establish a new individual fall within like 1 meter of the original parent, and probably much, much closer. That's actually explains why we see them the way we do in nature. Like they tend to be really spotty. So like one tree will have a lot of one thing on it, and then like the next tree will have almost none, and then like the next tree will not have any of that species. It'll have a different thing. So they're really sporadic, kind of like that. But what it also explains is why they're so sort of bad they suck at coming back, basically. Like, imagine it takes forever. These things do not grow quickly. So like they have to, like a spore lands a couple feet away from the parent, and then it takes like 20 years to like grow to be producing another spore. And then that's how the progression that you get. Occasionally one will move a really far distance, like the line, the like graph goes like this, and like this is the one spore that makes it like 100 feet from the parent, and even if it lands in a new place, it has to have that suitable environment, and most of the time it doesn't. So that's why it takes so long. There's this huge lag. And that's like something that is really difficult. So everything about lichens is so weird because you have to like detach yourself from reality and then like think about it, everything in a completely different way. We've never observed anywhere that's actually reached recolonization of lichens. Anything we're seeing now is like a process that is unfolding, but we don't know of anywhere that's actually completely, you know, reached the state that it originally was in prior to some kind of disturbance. And that's because we seem to think that these processes, like all the evidence points to that being a process that unfolds under millennia, you know, and like by the time you reach that point, it would have changed anyway.
So we have time for maybe one more question.
I didn't even talk about their reproduction. It's really crazy.
Hi. So I had 10 questions. No. Man. So I guess one big question, I'm not sure if you're going to be able to answer this, but when a lichen species evolves, does the fungi evolve? Does the algae evolve? Do they both evolve?
Is everything evolving all
exactly
at once?
Yeah,
everywhere maybe. Yeah, I mean the answer to that is evolution is occurring, and we don't have a really great idea of exactly how that's happening yet, because that partially has to do with we understand what's going on with the fungi because they're pretty easy to sequence, but then the DNA sequencing of the partners is more problematic. Like to understand the pool of all the partners in the thing is actually pretty hard. But yeah, they're both evolving. The question is like, so what I didn't, okay. The fact that these things can have these pools of partners inside of them tends to suggest that they're pretty flexible in their partnerships, right? So like, you expect in a system like orchids or like corals, where you have one thing and another thing, you expect really strong coevolution that results in really strict relationships. But lichens don't have that kind of specificity. So like an orchid can only associate with like one pollinator or whatever. And then if anything happens to one of them, the other thing is screwed, basically. But with the lichens, that fungus can associate potentially with a bunch of different partners. Those partners are just very closely related to each other. So like you expect that kind of coevolution to happen, but we don't quite understand what's going on the partner side in terms of like, are they really co-evolving with them? Or is it just sort of like that there's enough shifting around of partners that the lichen fungi evolve, but this evolution of the partners kind of happens independently? That can happen. Basically, it can happen every way that you can imagine. There's a really great review paper that was published in the New Phytologist last year that kind of goes into all of this, because really it's DNA sequencing that's sort of opened the box, you know, opened Pandora's box, so to speak, and made it clear that there's like so many options going on that like there's no one thing. Because that's the other thing, which I always forget to mention, is that the thing about lichen fungi is that it's a lifestyle. So it's a lifestyle that's evolved many times across all the fungi. Whereas like if you talk about sunflowers or corals, that's one lineage, evolutionary lineage of life. Whereas lichens are lots of different fungi that have evolved this lifestyle. Some are even losing it, but they've evolved it at different times. And they're like along various stages of the path towards becoming a real lichen. Like some of them are more, some of them are less. Sorry.
Let's thank our keynote speaker one more time.